All right, so we're going to pick up here with vesicular transport. So vesicular transport surrounds the idea that we are um, moving products, we're moving things into and out of the cell using a, a membrane enclosed vesicle. Okay, sometimes these vesicles can come from outside of the cell and fuse with the cell to put their products into the cell. They can also come from the inside of the cell, fuse with the cell, and put their products into the outside of the cell. And there are different variations of these, and we'll talk about them. So endocytosis is the general process of bringing something into the cell. So the steps are you have a packaged something out of the cell, or you just have something free. It can then sometimes bind to a receptor, or it doesn't necessarily have to bind to a receptor. It can just be brought in. It is enclosed within a vesicle, and then that vesicle, once on the inside of the cell, kind of pinches off from the rest of the plasma membrane and becomes its own separate membrane enclosed vesicle, this little sac. But what it's surrounded by, this sac, what's making up this membrane, is a portion of the plasma membrane. Okay, this will make I think it'll make a little bit more sense whenever you look at a picture of it. So types of endocytosis is phagocytosis. This is this idea of cell eating. So sometimes there are phagocytes, macrophages, and things like that in the immune system that will bring in and phagocytize foreign debris, foreign cells, old dead cell parts, things like that. Bring them in, and then oftentimes this will fuse with a lysosome and it will degrade and break down whatever it is was phagocytized. Pinocytosis is this idea of cell drinking, so bringing in water, bringing in fluid from the extracellular space to the intercellular space. And then receptor mediated endocytosis has to do with the plasma membrane being lined with receptors, and then some ligand or some substance or molecule will come and bind to it, and that binding triggers the process of it being endocytosed into the cell. And again, I'll show you a picture that'll make more sense. Now, exocytosis is exactly the opposite of that. It is when something from the inside of the cell that is packaged within a vesicle fuses with the cell's plasma membrane. Once it fuses with the cell's plasma membrane, it begins to open up, and then it will expel whatever contents it has to the extracellular space. Okay, so sometimes we may make a protein, it gets packaged into a vesicle, sometimes at this organelle known as the Golgi or Golgi apparatus. Once it gets packaged there, it can then be expelled once it fuses with the plasma membrane, expelled to the outside of the cell to the extracellular space. So here you can see some examples of endocytosis. Phagocytosis, you're bringing in something, so maybe this is a bacteria, maybe this is something else that gets packaged and brought into this vesicle. This is now called a phagosome. This phagosome can then fuse with something called a lysosome, and we'll look at lysosomes that contain digestive enzymes. When a phagosome and a lysosome fuse, you get something called a phagolysosome, and that's this phagosome that has fused with the lysosome. It dumps its lysosomal enzymes into the same area, and it will degrade whatever this little piece is. You have pinocytosis, this idea of cell drinking, so bringing in fluid into a membrane enclosed vesicle. And then for receptor mediated endocytosis, as soon as the ligand or as soon as the molecule binds to a receptor, that triggers the invagination of it, and it triggers it becoming enclosed within part of the plasma membrane, as you see here. So with all of these, for phagocytosis, pinocytosis, and this receptor-mediated endocytosis, all of these enclosed vesicles, you can see, originally came from part of the plasma membrane. They're just a section of that was pinched off and expelled to the intercellular space within the cell. So, and you can imagine if one of these was coming up, one of these vesicles, imagine this was coming up, it would fuse with the plasma membrane, and if it expelled its contents, in that case, that would be exocytosis, getting rid of something, putting it outside of the cell. Okay. Okay, now the next idea surrounding cells surrounds the concentration of different solutes relative to something else within uh, or something else outside of the cell. So you could have a glass of water or something, or a region of water, and then if you put a cell in that has a different, what we're calling a different tonicity, a different concentration of solute, something will happen to that cell depending on the concentration. So if a solution is hypertonic, meaning that it has a higher concentration of solutes than the cell that is put in it, that is called a hypertonic solution. There's also a hypotonic solution. And then whenever the cell and the region that it's put into, when they have equal numbers, proportionally equal numbers of solutes, that is called an isotonic uh, concentration or isotonic solution. So depending on what a cell is put into, it will react differently. So let's take a look at these. If a cell is put into a hypertonic solution, so imagine you have a glass of water and you have lots and lots and lots of salt. You put lots of salt in that water. And then you have a cell that only has a little bit of salt, much less salt relative to how much salt is in the glass of water. So when you put that cell in there, there is a higher concentration of solutes or salt within the water than there is in the cell. So what's going to happen is water is going to leave the cell to help equalize the concentrations relatively. So if you add more water to something, you're going to make it more dilute or less concentrated. So that's what's happening. You're taking fluid, you're taking water out of the cell, going to that extracellular space, which is the water that it's in, and that will cause the cell to shrink. So you can imagine if you sucked all the water out of a cell, it would shrink down. And that process is called crenation. Okay, so water loss leading to shrinking in a hypertonic solution is known as crenation. If you put a cell into a hypotonic solution, so one that has very little solute, and the cell itself has more solute proportionally than the water, then water will leave the glass, go into the cell, and you can imagine if you have so much water rushing into the cell, that's going to cause the cell to burst or to lyse. Okay, and then if you have equal concentrations of solutes relative, the cell relative to the glass of water it's put in, there's going to be no net change. Okay, there may be water come in and water go out, but you won't see any crenation, you won't see any lysis of a cell. Okay, hemolysis here, if I go back for a second, hemolysis is referring specifically to the rupture of red blood cells specifically. So here you can see this here. So in an isotonic, some comes in and some leaves, and it stays its normal shape. In a hypertonic, it's going to shrink and shrivel and crenate, and if it is in a hypotonic, it will get very large. One of the ways I like to remember this, isotonic is easy to remember. No change happens. Hypotonic, I think hypo sounds like hippo, and hippos are large. So when it's put into a hypotonic solution, the cell is going to become very large and will potentially lyse. Okay, we are going to stop there with this short little part, and we'll pick up with the next part talking about the electrical phenomena of cells.